One of the things that I'm somewhat guilty of asking folks is I often think of the mRNA and LNP. I like to ask folks if they go together like peanut butter and jelly, or peanut butter and banana, or peanut butter and banana, or peanut butter and marshmallow fluff, right? And so I'm curious to get your thoughts on how we ended up here today, right? We've had massive success with the mRNA vaccine so far. They were delivered with LNPs. But, in addition to the real world experiences that we have, how is it that LNPs won the carrier of choice today, if you will? And Russell, I'd like to start with you, thinking more about some of the scientific discoveries, perhaps, that have launched us to where we are today, that have created that framework for us, right, to work within. And then Aalok, I'll turn it over to you to talk a little bit more on perhaps the CMC side of things. So, Russell, what are your thoughts?
Well, thanks for asking me the question, Anna. It's a real pleasure to be here. Certainly, it's an exciting topic, a topic that I'm really excited about, and I think we'll have a great time talking about the role and the impact of mRNA vaccines and therapeutics. Certainly, I think there's a lot to discuss. Also, thanks to PNI for hosting, and it's a pleasure to be here with Aalok to get to know him a little bit better, and really as we kind of build and connect across the mRNA delivery world. So, I think there's a couple of key innovations, really. Certainly, kind of the cationic lipid is where most people would start that story. And a lot of those innovations were really driven, by UBC, the Colas Group, the Colas-related companies, and then their partnership with Alnylam, really, and how they drove forward, delivery with siRNA, and how that really was further, enabled by a lot of other companies that were working in that same area. And I think really the key innovation was really that they could map out kind of a structure and function relationship of cationic lipids, really identifying key features of those lipids, like the PK of the head group, the branching structures, and the kind of the number of tails that you could really demonstrate with increased potency. So that's where I think, in my mind, that this area of delivery really began for lipid nanoparticles.
Mm-hmm. Mm-hmm. Awesome. So it's about sort of the level of transparency we had into it, right? As a molecule, something that can target how it can target, what it can and should look like analytically, right? That kind of helped map where we are today.
Yeah. I mean, I think you could, you could assign some function to one component, right, of a lipid nanoparticle, right? And that was the cationic lipid. I think there was a lot of other, really deeper research and understanding around lipid nanoparticles that really have come, I would think, more recently. But really, I guess it's two things. One is really the field needed to learn, what was potent, what wasn't potent. But then also the field needed to learn how to put a nucleic acid really inside of a particle, right? And that didn't really happen efficiently until really 2005. And that was, really, kind of pretty close to our modern process for making LNPs. So those are kind of two key things that really got the ball rolling. We really could put the mRNA inside of a particle, a lipid nanoparticle, effectively with high efficiency. And then we were able to really show that this was very potent, actually, in humans, to the level that it became therapeutically relevant.
Wonderful. Aalok, what do you have to add? I know one of the things you'd mentioned in our pre-call, right, is that there are a couple of other CMC reasons, too, behind why these two make such a great pairing. What are your thoughts? Do you have anything to add to what Russell has shared?
Yeah, sure. First of all, thank you for having me as well. This is a great forum, very enlightening for a lot of people and for us as well. But yeah, CMC, so, when I think about a product becoming and maturing into, actual human clinical use, right? You need scalability, and that's where the CMC aspect comes in. And nanoparticles are nanotechnology, as we used to call it, these little tiny blobs. It can be PLGA, liposomes, all these things, right? If you look at the history, liposomes, for example, are pretty famous. Doxo is an example. And the process has evolved, right? So the LNP, the vaccine LNPs, classic mRNA or oligonucleotide LNPs, are even more simpler in process when it comes to scalability than liposomes. Because liposomes, you have to do active loading and all those sort of things. Here, it's just a simple T-junction process or a chip process, whatever you choose, right? And the simplicity of the process makes it much easier to scale, right? And so when millions or even billions of doses were needed for vaccine production, right, it was quickly brought online 2 years ago, as everyone remembers, and that's the key, right? Whereas in other advanced therapeutics like monoclonal antibodies and whatnot, they're cell-based, and it's a little more complex than this, right? So those aspects are really important when in contributing how our technology matures, right, and is widely available for use.
Mm-hmm. Absolutely. Scalability is the topic of the day in pretty much every sector of the advanced therapy industry, right? It's what we're all challenged by at the end of the day. And so we've really had a great example from the pandemic, right? We've learned that we can scale up mRNA LNPs for a large vaccine population.